that will be moderated by Mark Ferrer as well as Nikisa Sadre. So I think we'll have all of our speakers come up here, please, as well as Mark and Nikisa. You can, no, yeah, you can sit at any of the tables anywhere. Because they all are handheld mics. I mean, the mics all work. So, perfect. Perfect. Oh, I see. Good. Okay. I want to start out with just two questions, and then I'm going to let the facilitators continue forward. The first question comes from the audience. Drug development tools generally generate comparative risk profiles across compounds rather than absolute clinical prediction. What level of concordance with known clinical risk ratings is sufficient for qualification, and how should developers communicate the distinction between ranking accuracy and predictive precision? Just going to start out with a hard one. So, who would like to start? Jonathan?
Sure. So in many ways, I think that is a question for the drug developers. But the first thought that I have is it really depends. Again, the context of use. Is this on? Can you hear me? Oh, you have to get really close to the microphone. Okay. So I think it's a question really for industry in some ways because it's the risk tolerance and development of the drug. But it depends on if you have a disease that's life-threatening and there is no therapy, and the only therapy is highly toxic, but it's currently in use because the risk-benefit ratio for a toxic therapy, it may still be beneficial if it prevents a complication that's worse than the toxicity from the drug. And if you can have a drug that is still toxic but is only half as toxic as the drug that's in clinical use, that's an advancement. So, we live in a world where most of the drugs that are in use have some toxicity. They can be idiosyncratic with, certain individuals get a complication and it's not general, or it can be a known complication of the drug. But again, if you're treating for example, cancer therapeutics or infectious disease therapeutics, you want to pick the less toxic but equally efficacious, or the equally toxic but more efficacious treatment. So it really depends on, I think, on what the indication for the drug is and what the current alternatives therapeutically are. That would be my first response to that. First thought.
Anybody on this side?
We haven't put a context of use in for that yet, but we did do a study where we worked with a company, Vanda Pharmaceuticals, where we took our four organ system and utilized that to replace a 9-month Beagle study to be able to get their drug under review, which enabled that. And what we did there was we actually took their drug at different concentrations with different liver phenotypes, okay, and then did 2 positive controls for each drug system. So from a context of use, it doesn't always have to be comparative in terms of a risk benefit across a continuum. You can also just have, actually known toxicities for each organ system, okay, and then compare that to that drug. So it doesn't have to, in my mind, always have to be in terms of a continuum in terms of the risk compared to other things. You can actually then take it, okay, this actually system shows you can actually predict this toxicity. What is that in comparison to the actual drug you're looking at? So that was another take on how to do that.
Yeah, I wouldn't actually get hung up on trying to replicate the full clinical syndrome in a dish or, on a chip. And for some of the conditions, as Mark was showing, if you can relate, steatosis and fibrosis and inflammation to the clinical syndrome or classification, that's fantastic. But I think for most of the clinical phenotypes that we're trying to avoid, we will have to rely on some form of a mechanistic readout. And we obviously have to have a considerable comfort that when we see a cytokine or a cell death or other readout in the cell, relating that to the ultimate outcomes. And that's, being done in risk assessment all of the time. We don't have to have the animal study to label a chemical carcinogenic. We can rely on DNA damage and genotoxicity. So I think it will be context dependent, but I don't think it's reasonable for us to expect to recapitulate the clinical syndrome in a dish, and we'll have to understand very well the mechanistic biomarker and use those readouts. And everything ultimately will be put together in a weight of evidence approach anyway.
Nikisa or Mark, did you want to?
I just, in the context of qualification, I think to me that the discussion this morning is qualification, right? Whether the need for a competitor, the compound with known clinical effects, like whether it's toxicity in the kidney. You mentioned, and I think it was mentioned that amyloid has a comparator in the DILI as well. Do you need that competitor for qualification? And one thing is the use of a model for early discovery or within internal decisions, but from the context of qualification, it looks like you need a competitor. You need a compound with similar structure with known information in clinic. Is that something that you guys discussing with ISTAND or critical path? Is this a must-have for your qualification?
Nikisa?
No. You need to be able to you can use positive controls, negative controls. You don't need a comparator. It's not a requirement. I think in the case of the two LOIs, I think that because specific class of drugs was being evaluated, there were comparators to use, and so I think that was made part of the context of use, but it's not something that's required, for qualification. And I also want to highlight that qualification is not required in order to be able to use your model in a drug development program. So I think that while it's good to qualify, I think that it sort of increases confidence and from what I heard from the presentations today, I was happy to hear that there was an educational component involved, which goes in both directions, which I think that, is often been the emphasis that, FDA needs to learn about these things and needs to learn about these, how these models are, performing and everything. But I think that developers also need to understand drug development and what, how you are supposed to use these models in drug development. And so I think that there's this kind of educational aspect has been very, a positive result of this, because people kind of showed today lessons learned, which I think are going to help in future, sort of packages that are submitted. And one other thing that I just wanted to mention, maybe it's related to this topic, obviously, but this question, which I think Ivan also kind of brought up from a slightly different terminology perspective. I think that we need to separate the sort of risk assessment from hazard evaluation. And I think, when you're looking for very discrete things, it's mostly hazard identification. And so these methods are, looking at individual endpoints, and those are individual hazards. You then have to put them all together to evaluate a particular risk. And so, that's an important aspect to keep in mind, that you're not going to get rid of an animal study or radically change something just by at knowing what a couple of hazards might be, unless you understand the entire slew of hazards associated with a particular pathway. And so I think, for example, for particular models such as like skin sensitization, where the whole adverse outcome pathway was able to be broken down into steps, and each step was kind of like addressed with different types of in vitro models for evaluating it. That's kind of illustrates how you need to break down these things. And so it's not a single model that's going to, result in you being able to de-risk something altogether. You're just kind of answering, and that comes into the weight of evidence approach. But anyway, I hope I addressed a number of questions.
Thank you. Nick?
That question made me have three thoughts in my head. First one was, I need another coffee, because that's a pretty hardcore question.
Right.
Second one was, I mean, that's really important, but there's an old show called Kung Fu where there was like this guy being trained, and he said, one must walk before one can run, grasshopper. And I've been doing this for about 25 years now, since I was an undergraduate. And yeah, we made a ton of progress, but that's a really complicated kind of use case there that I think we might be ready for, but I don't think that's like the state of the art right now, the technology. But there are a lot of really good questions that we can answer with this tech already. For example, your example of, we can refine compounds in concert with an animal study. So for example, we had a customer who had like 5 gene therapy candidates, and they're like, I can only afford to do 1 or 2 of these. Which one should I do? And then we tested it, and rather than doing a $3 million 9-month mouse study, we did in 21 days. We're like, okay, these are the 2 that you want to go with. Or like, in a couple cases, we've lost customers, and we've lost them because their compound just didn't work. And we're like, yeah, okay, stop spending money on this. And that's also a good outcome for us too. But as we saw from the BoPatch, the Botox one that we're working on, the botulinum toxin, that's a pretty narrow context of use, which is really fortunate, because how potent is this? That's a major, major problem for a lot of drugs outside of these toxins. Like a lot of gene therapies for muscular dystrophies have been put on clinical hold the past couple of years. And we helped a couple customers get off of that clinical hold because their potency assays they're non-functional. They're not using functional data. It's like, okay, is my protein there? Great, the drug works. That's not the whole picture, right? So is it causing muscle contraction? Yes, that's really the level that we get to. And then the third part was, there are times when we don't have the luxury of that. So, we work with a family, the Corman family in Texas. They have a son named Austin. He's like, I think he just turned 7. He's got a disease that maybe 50 people in the world have the same mutation as him. And so there aren't really a lot of options as far as like testing different medicines and having different models and having the luxury of answering those types of questions. And I know these are edge cases, but we've been talking about this fantasy of N of 1 clinical trials or N of 1 medicines. That I think is a pretty nice use case for these types of technologies that kind of go outside of being able to answer the scope of that question that Pasley started with.
Excellent. Okay, I think we can go to our second question. Our second question is, academic developers often have mature platforms, but very limited visibility into the drug development gaps that those platforms could fill. Beyond securing a pharma partner, what other mechanisms, such as pre-LOI consultations, transparency database review, facilitation through nonprofits such as CPATH, are most effective for helping developers position a scientific capability to address a regulatory need?
I mean, there are opportunities to meet with FDA. There's a sort of mechanism under the Interact, I think it's called Interact meetings. And so for a situation like that, which is not associated specifically with an application, if you have a drug application, then you can come in for a pre-IND meeting and have a consultation basically and sort of provide your thoughts on something. But there are, with the Interact meeting, and then there's another one, I think a CIVM meeting as well. Mm-hmm. And these are types of meetings where you can meet with subject matter experts and regulatory sort of experts to try and explain, your platform and have some guidance as to what might be approaches that you might use to sort of, enhance your chances of being able to apply it. And so those are opportunities which are available, and, we hope that people can use them. I don't know that they use them as much as maybe they could, but I think that it would be good if people actually, submitted requests to, meet with FDA in order to present these things. It's also an opportunity for FDA to become knowledgeable about some of the advances and some of the new technologies. And so hearing it directly from the platform manufacturers would be very helpful, because I think, a lot of industry, they know what they're doing. For them, it's different. But for academic developers, they probably need a lot more advice, both from the drug development perspective as well as from the regulatory and sort of context of use perspective. And so I think that would be a good sort of scenario for having academicians meet with FDA. So
Wonderful. Nick, did you want to follow on?
I feel weird commenting about this because I'm not an academic, but we're a small startup company spun out of the University of Washington, so we've got a lot of academic contacts. One of the really helpful things for us was actually came from FDA and NIH, and that was the work that we presented today was part of an SBIR. And Sashi Sharma, who's in the back, his life's work has been to replace the mouse lethality bioassay for these types of studies. And had there not been that kind of confluence of industry need and developers like us who know how to make these things commercial, know how to make these things easy to use, we have that expertise. But also having folks at FDA and NIH saying, come together, put these two pieces together. Here's the need. Here are the people who can solve the need. Let's make it happen. That was absolutely critical because I really I'd say I don't want to put a timeline on it, but that we're not too far away from botulinum toxin never being tested on animals again. And we have a customer who uses over 1 million mice a year just for potency testing, and we're working with them to replace all of their work with that. So again, FDA, NIH were absolutely vital for that part.
Anyone on this side?
I'm going to make a shameless plug here right now for the MPS meeting. I don't know if you know, the International Microphysiological Systems Society been holding annual meetings. Okay, next meeting is going to be the end of May. Okay, in Washington D.C. Okay, there's a chance for all your academicians to come in, look and see what people are doing, talking to the people who are making these systems. There's going to be a huge regulatory presence there as well. So this is a way for you to really go in and judge where you stand with your system in terms of all the people out there, in terms of commercial activity, but also other academicians and also regulatory folks. So again, if you weren't aware of it, please think about it. Abstract submissions are still open. It'll be a poster, it won't be an oral, but you can actually get that in there as well. So if you want a form to be able to get and understand, what your options are, that is the perfect place to go to.
Thanks. Ovan?
You'll hear from Dr. Sokolish later today on our tissue chip testing efforts, but in the last 10 years, we've seen probably most of the academic and commercial platforms in our tissue chip testing center. And when someone says there is a mature platform in academia that is not getting any traction, this surprises me because I haven't seen a mature system in academia. And again, this goes to our own colleagues at Texas A&M. They always are, trying to improve the system, materials, manufacturing, multiplexing, whatnot. But the bigger challenge for academicians who think that they have a mature platform is to think whether that mature platform is needed or not. I can tell you that there's plenty of pharma and chemical industry users at every toxicology, disease or MPS meeting who are looking for solutions. So if you have to ask yourself a question, what problem are you solving? Not whether your platform is mature. So if you can answer both of those questions, customers will be killing over in trying to get your platform. If they're not, then perhaps your challenge is not solving a problem that actually needs to be solved.
Yeah. Oh, Jonathan.
So I'll make a slightly different shameless plug than Jay's plug. In the early days of the NCATS tissue chips program, the IQ-MPS consortium came to every meeting. Pharma, biotech were very well represented, and there was a lot of interaction that took place, and it was very, I think, on all sides, that was very helpful. Maybe something NCATS and/or CPATH could do something like that, convene, and maybe it could be at the upcoming MPS meeting and other forums too, but it is good to get the academic developers and the industry representatives that have a particular interest in MPS systems, not just the whole company, because a lot of these companies, the left hand and the right hand, don't even know what they're doing. But you get the people who are particularly interested from biotech and pharma in the room with the academic developers or just in the hallways. And good things happen on both sides. You learn more on the academic side. You learn more what you have to do to make your system viable and useful from an industry perspective. So maybe reconvening some of those or creating opportunities like that in the future would be helpful.
No, thank you. And then Dr. Rebecca Pompano, you have a question? Oh, it's not. Is it on? It's not on. Anybody? And let me see if I can. Hello? Hello? This one works.
Great. Okay, great. Thank you. I really appreciate all your expertise up here. So something that I was wondering about is in terms of maturity, how do you think about reproducibility? Are there guidelines as there are for analytical tests, the more conventional chemical analytical tests in terms of coefficient of variation that's required? And how do you think about that within, for chips that use primary cells, especially ones immune related that are using, primary blood derived cells? How do you think about the reproducibility within a donor versus between donors in terms of regulatory use?
I mean, you're touching upon the sort of concept of validation here. And, when I said qualification was not necessary, that I meant that, but validation, unfortunately, is going to be necessary. But the validation doesn't have to be formal validation where, you kind of have to do it in multiple sites and have a sort of a standardization organization kind of like validated. But there are certain validation criteria, and I think, CIVM has published on what some of the, criteria are. We tend to agree with a lot of what's in there, and a certain amount of validation which addresses context of use, biological relevance, technical characterization, fit for purpose are the types of things that are going to be necessary for any type of platform that's submitted. And within the technical characterization, reproducibility, sensitivity, specificity, all of these types of things are going to be, necessary to describe so that there's confidence in the methodology. So you absolutely bring up a good point that once the method is sort of identified and you kind of have a context of use, you still have to go through a process of demonstrating that it actually works. And so that's one challenge that we understand is obviously real. We have a partnership, a public-private partnership with the 3Rs Collaborative, where we're kind of looking across liver platforms and at what the level of variability is if the actual platform manufacturer were to do the test by themselves using the same protocol by all of the sort of members of the partners in that project. What would be the variability seen? Because we anticipate that some variability has to be acceptable. So I guess what we want to know is that, if a sponsor comes and submits data from one platform versus another platform, and these are not equivalent results that we see even with the same positive and negative controls, then what are we supposed to make out of that? And so I think these are things we need to get a handle on, but I think that there's going to be some room for flexibility, obviously, but that's an important, I hope I addressed the question.
Well, brings up a really important point about what statistical approach do you use for these systems, right? So there's one thought is that, well, you're trying to create an assay, so enzymatic assays and things like that, single cell assays, well, maybe you need to use those statistical aspects. But what if you're trying to replace an animal? Animal experiments have very different sets of statistics, right? And so you'd think if you're trying to replace an animal, that the same statistical methodology used for animals should be actually applied to the MPS as well. So I think that's actually a discussion that's going on right now, which is what statistical power do you need to, apply to these model systems, okay? Because if you're trying to replace animals, it should be different than if you're trying to do something like an enzymatic or a single cell reproduced assay. So I think that's something the field really needs to consider at this point in terms of what statistical methodology we use for these model systems.
Mark?
So maybe it's a shameless plug too, but we have developed the Pittsburgh reproducibility protocol to address this specifically for the MPS, where we have a series of statistical tests where you can go back and see, well, how reproducible is, our model at this level, at the absolute level or at the relative level. So we're trying to, we've introduced what we think would be a standard here. But I think we also have to realize that the difference between reproducibility and heterogeneity, and that would be reproducibility if you're using the same cells, the same protocol, everything. Then you're testing about reproducibility. But you start changing, okay, I'm going to use this patient's cell instead of this patient's cell. Well, that's no longer reproducibility. That's an assessment of the heterogeneity. And so that also has to be taken into consideration.
Thank you. And then do you have a comment, Yvonne or Jen?
Just because you called on me, I will. I mean, the patients are heterogeneous too, right? So there is a donor variability that's actually capturing the clinical spectrum. And I think sorting through that, being able to isolate it as that is a feature and an advantage of MPS, or it can be. And so we have to find a way to make sure we're isolating, something good, variability that's real, from reproducibility, right? So just a comment, yeah.
Thanks. And then Yvonne?
And just quickly, they're really practically, despite all the guidance out there, good in vitro laboratory practices, they're voluminous texts that really are not being used. I think the two things we've seen that the end users are most interested in is the standard statistical tests of sensitivity, specificity, and overall prediction if you have a phenotype. And the best example I'm aware of is the work by Emulate, in their DILI chip. And 28 compounds was a gargantuan effort. And really, you need hundreds to really have confidence. So that's as a road that you can only go so far, given the cost and the throughput of your model. But what we have found end users really need is they need to understand how many replicas they need to run. And therefore, in our TaxVal consortium, almost every paper includes the retrospective power analysis. Once we run the experiment a number of times, then we can actually reanalyze and say for basal function to see differences, you need X number of replicates. For some severe effect of a positive control, you may be okay with this number of replicates. Those parameters are really most useful, and I would recommend that developers, as they do more and more experiments, kind of take a retrospective look at their power.
I think in terms of time, we may only have time for about one question, so.
All right, I'm lucky the last question. So I really want to ask about the clinical dosage. So usually for the MPS system, the dosage, how the gap will actually compare to the real clinical dosage, how to manage to really choose the right dosage when running the clinical trials if the data come from the MPS system.
Who would like to start?
Well, as I presented, we've already published a digital twin that did exactly that and came very close for 3 drugs, antimalarials, to what they actually do in the clinic. Okay, for maximum tolerated dose, no adverse spectral level dose, and EC50, based upon the dosing regimen. So that's already out there, but it's going to be a combination of in silico and MPS to be able to get to there, in my opinion.
Jonathan?
You can do pretty sophisticated PBPK modeling and in vitro to in vivo scaling in these systems. It is a lot of work to do, but we've done that and others have done that as well. And that gives you much more confidence that you're of course using a range of doses in your models, but it gives you much more confidence that you're capturing the likely clinical exposure, at least short-term clinical exposure, to be able to model from in vitro to in vivo. So those systems do exist, and they have been used in some of these MPS systems.
So that means for running the clinical trial, probably still need to try the dose finding at the beginning in the trial design.
So one of the potential contexts of use for some of these systems is to help define the initial dosing for clinical trials. And that, for example, we are preparing a second letter of intent where that will be part of the letter of intent in a kidney organoid model for picking the appropriate doses for initial dosing in clinical trials. So I do think these systems do have utility, potential utility for that.
All right, thank you.
Yeah, Nick had one
I'll
more
be
comment.
brief, kind of a hybrid of the two answers that you heard here. We've seen, it depends a bit on compound class. So some compounds, it's actually almost a one-to-one translation between dosing in the dish and dosing in the patient that we've seen. But then in others, we have to do a bridging study basically to find out like what's the conversion factor. But every once in a while, like, for gene therapy studies, right, for certain AAVs, we see it's almost one-to-one, where we've like replicated some clinical trials where like kids have died due to liver toxicity and stuff like that. It's like, yeah, we could have seen that quite easily. So I don't think there's a one-size-fits-all answer based on compound class, what's the indication and stuff like that. But yeah, it's really valuable to do that, and we're looking at doing like clinical trial screening of individual patients and stuff like that to see if we can get advanced insight before you start putting it into people.
Well, thank you all so much. I want to thank again our speakers as well as our session moderators.